right. Hi, welcome to Lecture 2 of Abnormal Psychology. Today we're going to be talking about the cognitive behavioral model, which is an example of a modern integrative understanding of mental health and mental illness. In the last lecture, oh, hello. All right. In the last lecture, we went over the history of different understandings of psychopathology, different models that people use to explain human behavior and human maladaptive behavior. And towards the end of the lecture, we made a distinction between unidimensional models, which are explanations that really focus on one mechanism, one reason why behaviors develop, and one reason that maintains behavior. So, for example, ghosts, the devil in the spiritual model, society in the mental hygiene model, or learned associations and conditioning in the behaviorist model. And in the last lecture, I contrasted that with multidimensional models of psychopathology and emphasized that because human behavior is really complicated, one model, one causal explanation really can't explain all of psychopathology. We need to take into account biology and physiology, as well as cultural context, someone's role in their society and their community, so someone's social relationships, and as well as developmental models. So not just what's happening to someone right now and what their social context is right now, but taking into account what their social context was and what their interpersonal relationships like when they were growing up. It's not in the outline because we're really not going to focus a lot on it in this class, but the cognitive behavioral model also takes into account humanist ideas, especially the idea that, as we talked about last time, people need to have respect and dignity as well as safety and physical security. And it's not always talked about in abnormal psychology, but it's something that we think a lot about in clinical psychology more broadly, and especially as therapists treating individual patients. So it's important not to lose sight of that. So today's lecture is going to start out by defining unidimensional versus multidimensional models of psychopathology, and then we're going to take a big detour into talking about neuroanatomy and brain structures that are important in psychopathology. So the book goes into this in a lot of detail, and I'm going to sort of try to review most of what the book covers while highlighting things that are going to be the most important for this class. Okay, let's get started. So unidimensional models are models, as I said, that explain abnormal behavior in terms of a single cause, like spirits or learned associations. As we talked about in the last lecture, behaviorism, which really focuses on learned associations as the single cause of both healthy and maladaptive behavior, can really explain a lot about psychopathology. So it's a really important and promising model of behavior. But even behaviorism, which explains a lot, can't explain everything. So just some more examples for you. In psychoanalysis, the unidimensional aspect there is the idea that personality is the result of fixations that happen in specific developmental stages. Whereas, as I said, in behaviorism, the unidimensional model is that emotion and behavior are kind of caused by reinforcement and learned contingencies in the environment. So one thing that behaviorism doesn't do the best job of explaining is, for one thing, where unconditioned behaviors come from, why we have natural unlearned reactions to certain things in our environments. So we'll talk a lot about that in the biology and evolution section of today's lecture. It also, because behaviorism is based on animal models and on sort of finding analogs in animal behavior to human behavior, it's not the best at explaining behaviors that humans don't share with animals. Things like consciousness, self-concept, self-esteem, are humans' ability to think about and predict the future, and human culture, the fact that humans have really complicated, deep social relationships that really influence our lives and behavior. Behaviorism alone can't explain all that. So a model that makes an effort to explain all that, to take all of these sort of evidence-based scientific theories into account in explaining human behavior, is cognitive behaviorism. And cognitive behavioral models are really going to be emphasized in this class. Mostly cognitive behavioral therapy is a form of treatment. But as I mentioned in the last lecture, every form of treatment is based on an understanding of cause. And so is CBT, cognitive behavioral therapy. It's based on an understanding of psychopathology is rooted. Okay, but as I said, first we're going to talk a lot about neuroanatomy. Okay, so the basics of neurobiology start with the central nervous system, which is the brain and spinal cord, and the peripheral nervous system, which is how the brain and spinal cord communicate with the rest of the body. The peripheral nervous system has two divisions, so there's two branches of the peripheral nervous system. The first, the autonomic, sends orders out from the brain to the body. So these are things like voluntary movements, like moving our arms and legs to walk and run, as well as involuntary movements, like moving our diaphragms to breathe or moving our organs to digest food. The other branch of the peripheral nervous system is the somatic branch. That branch brings information in from the body into the brain. So it's how we sense things in our environment. It's all five of our senses, as well as our sense of where our body is in space and our sense of physical movement of the body. So the peripheral nervous system has two branches, as I said, but the autonomic nervous system is further divided into the parasympathetic and sympathetic divisions. And the most important thing to remember about the sympathetic nervous system is that it's the nervous system, the branch of the nervous system that handles fight or flight. So when we're threatened, when we're feeling endangered, the sympathetic nervous system takes over, and it does things that prepare the body, prepare our physiology to either defend ourselves against a threat or a predator or run away. So an example is that when the sympathetic nervous system is activated, it increases our heart rate. And what that does is it helps to send oxygenated blood to our limbs. It also does things like slow down the digestion, inhibit stomach activity, so stop all of the gastric contractions that are happening as we passively digest our food. And it does that because slowing the gastric contractions and stopping digestion frees up more blood to go to the rest of the body to help us run away or defend ourselves. By contrast, the parasympathetic nervous system is also sometimes known as the rest and digest nervous system. It's the branch of the nervous system that is in control of our movements most of the time when we're feeling safe or neutral, not under threat. And so the functions that are controlled by the parasympathetic nervous system are things like normal digestion, regular steady breathing, bladder contractions, so controlling our bladder, and digestion. So this is just a little figure for you guys to study. If you have any questions about just the basic divisions of the nervous system, please ask them in Thursday's class or office hours or one of the in-person classes. Okay, so the brain and the nervous system is made up of nerves. Nerves are a specialized type of cell. They are primarily in our brain and spinal cord as well as in our peripheral nervous system to communicate with other types of cells that are in the body. So what makes nerves unique and special is that they communicate with neurotransmitters. Neurotransmitters are chemicals that nerves release through their axon. So this is the end of a nerve. This is the cell body of a nerve. If you've taken biology, this is the part of the nerve that has the nucleus and all of the important cell membranes that make a cell. This is the part of the nerve that's really specialized for a specific kind of communication that's mediated by nerves. So nerves receive electrical signals at their dendrites. Nerves receive chemical signals at their dendrites and convert them into electrical signals called action potentials that are propagated down the axon and cause the release of the neurotransmitters that neuron produces at the axon terminal. So basically nerves, their function is to translate chemical signals into electricity back into chemicals. nerves communicate with other nerves by producing chemicals called neurotransmitters. It says right here neurotransmitters / hormones because chemicals that cells can use to communicate, which are produced outside of the central nervous system, so in the body and the organs, are called hormones. And those chemicals, when they're produced within the central nervous system, are called neurotransmitters. Okay, so let's talk a little bit about the structure of the brain. Most of what we're going to talk about in this class, almost all of it, happens in the forebrain. So this is the more evolutionarily recent part of the brain. It is the part of the brain that humans and mammals share, but that some lower organisms like nematodes and other kind of small insect-like organisms don't have. So we'll talk about the divisions of the forebrain a lot more, but the main parts of the forebrain that we focus on are the cortex, which is the outer layer of the brain. It's the wrinkly outer surface of the brain that contains things like, it's where things like memories and learning are stored, and also where we integrate and process stimuli from other parts of the brain. The limbic system is an evolutionarily old structure that sits closer to the middle of the brain. And it's really involved in things like appetitive drives, so hunger and thirst, as well as sex and aggression drives, as well as emotion and pain sensations. And then the basal ganglia is, I think we mentioned in the last lecture, in the context of deep brain stimulation. It's also a deeper brain structure where dopamine is produced. And dopamine is a neurotransmitter that is, among other functions, really important for voluntary movement. So Parkinson's disease involves damage to the basal ganglia, causing it to stop producing as much dopamine, which has a first impact on voluntary movement. So in addition to the forebrain, we have the brainstem, and I'll show you a picture of this on the next slide. But the brainstem contains two divisions, the hindbrain and the midbrain. So the hindbrain is really what regulates autonomic functions. So the things that passively keep us alive. Muscle movements, so autonomic nervous system involves the brain directing the body to move, but things that we don't use conscious thought to control. We don't need to remind ourselves to breathe. We don't need to tell our heart to beat. We don't need to consciously regulate our blood pressure. All of those things are done by the medulla. The pons is a hindbrain brainstem structure that regulates sleep. And that is actually one part of the brain outside the forebrain that we will talk about in one of our lectures. We'll talk about it in the lecture on sleep disorders. And then the cerebellum is responsible for coordinating between the somatic and autonomic nervous system. So in order for us to be coordinated, we have to tell our body what to do. We have to tell our arms and legs to move in a certain rhythm. But in order to do that accurately, we also have to be receiving sensory input from our arms and legs that tell our brain where those body parts are in space. Because if we don't always know where all of the parts of our body are, we can't coordinate the movement between two different parts of our body. So the cerebellum is part of that coordination. And then the midbrain also is responsible for coordinating autonomic signals with sensory input, so somatic signals. The midbrain and cerebellum are where the autonomic nervous system and the somatic nervous system are really integrated. All right, so that's a little bit blurry, but this is just a very basic illustration of those three divisions of the brain that I talked about. So the biggest part of the brain is the forebrain. It's this whole structure here, everything that's pink, and this right here, I think you can see my cursor. This is the limbic system, the structure in the middle. The midbrain is it sits at the top of the brainstem, so it's the part of the brainstem that's kind of surrounded by forebrain. And then the spinal cord structures are kind of right here in the back of our neck. So they're partially enclosed by our skulls, but they sit below the majority of the brain. Okay, so one of the most important forebrain structures for psychopathology is the limbic system. And broadly speaking, the limbic system is involved in emotions as well as drives. So Freud would have said, if Freud knew neuroanatomy, he would have said that the id lives in the limbic system. And in particular, probably the hypothalamus, which is the limbic structure that regulates appetite, thirst, sex, and aggressive behavior. But also the amygdala, which is the part of the brain that really is the most implicated in emotional experiences, and specifically fear and anxiety. So the amygdala is a structure in our brain that probably evolved to help our ancestors stay safe from danger, to experience an emotion that caused them to be motivated to run away, to protect themselves when they were in danger. And then the last structure in the limbic system is the thalamus, which is the part of our brain that's, among other areas, most responsible for pain perception. Other structures that are in the forebrain but outside of the cortex would be the hippocampus, which is the part of our brain that's really responsible for memory and learning. So the hippocampus is important for psychopathology too, because as we've talked about, and as we will talk about a lot more, a lot of psychological disorders are based on maladaptive learning. So learning feared associations that are unhelpful, or learning behaviors that can cause dysfunction, distress, and danger. And then, as I said, the basal ganglia coordinates voluntary movement, or it initiates voluntary movement by producing dopamine that goes to other brain structures that ultimately tell the limbs to move in conscious ways. That's different from coordination, which is totally unconscious. We don't have to think about making sure that our legs are moving in the right pattern to walk. In fact, if we do think about it and our conscious mind takes over, it can actually make us more clumsy. It's one of those things that is kind of always running in the background. So the basal ganglia is specifically voluntary movement, so motions that we're conscious of making, motions that we can articulate and say that we're going to do them, and we can explain how we're doing them, versus movements that we don't have to control and don't always consciously predict. So again, here is a diagram of forebrain structures. I didn't mention all of them because not all of them are super critical to understanding psychopathology and abnormal psychology, but this gives you kind of a sense of where everything is in the brain. Okay, yeah, the hippocampus is there. Cool. Okay, so probably the most important part of the brain, which is like a hard thing to say because the whole brain is extremely complicated and very interconnected, and all of it is involved in keeping you alive. But the part of the brain that's most important to your conscious experience is probably the cerebral cortex. So this is the part of the brain that is consciously interacting with the world. This is the part of the brain that mediates your experience of the world. And the cortex is divided into 4 sections. In the last lecture, we talked about the frontal cortex, the frontal lobe. So the brain is divided into 4 lobes, not just the cortex. But when we talk about cortical structures, this is the part of the part of the lobe on the outside. So the prefrontal cortex is the outer layer of the frontal lobe. And that cortical area, the neurons that make up the outside wrinkly part of your brain, are involved in lots of important things like planning, exerting self-control, and reasoning, things that we talked about in the last lecture in the context of lobotomies, which intentionally damaged this part of the brain. The prefrontal cortex is also the part of the brain that lets you think about the future and basically have an imagination. So imagine a world that doesn't exist, the future world. Also in the frontal lobe of the brain is the part of the cortex that mediates the sensation of smell. That part of the brain is the olfactory bulb. It sits kind of right behind your nose, but at the base of your brain. Also in the prefrontal cortex is the part of the brain that controls voluntary movement. So the dopamine from the basal ganglia is going to the prefrontal cortex, to the motor cortex, to produce voluntary movement. And that includes speech, which is a voluntary movement of the tongue, the palate, and the mouth. So the temporal lobe and the temporal cortex is the sides of your brain. And this part of the brain is really involved, for humans, the most specialized thing about the temporal lobe is that it is involved in language and speech recognition. The prefrontal lobe is involved in speech production because, again, that's a voluntary movement. But the temporal lobe is where we receive oral inputs, so we hear things. And if the thing that we're hearing is language, our temporal lobe recognizes it as language and allows us to understand it consciously. This part of the brain is also involved in visual recognition, both just general visual recognition and also a very specialized part of the brain called the fusiform face area that specifically helps us recognize faces, facial expressions, and recognize people that we know by looking at their face. Also in the temporal lobe cortex are areas where long-term memories are stored. So memories are formed in the hippocampus, but then sent out to different parts of the brain for long-term storage. And our memories basically exist as clusters of neurons that are constantly in communication with each other that are spread out throughout the cortex. So this is why amnesia, forgetting biographical details of your life, is a symptom of a stroke that happens in this part of your brain specifically because it affects long-term memory. So right at the top middle of your head is the parietal lobe. So whereas the motor cortex of the prefrontal lobe, motor cortex of the prefrontal lobe is involved in coordinating voluntary movement, the parietal lobe contains the somatosensory cortex, which is involved in receiving and interpreting and bringing to conscious awareness sensory information from the body. So again, we talked a lot about different parts of the brain that relay sensory information. It starts in the brainstem with integration of the autonomic and somatic nervous system with signals coming in from the body. They go to the brainstem first, then they're sent to the limbic system, and then from the limbic system, but also in a parallel process that happens at the same time, they're sent to the cortical regions. And one of the places they're sent is the parietal lobe somatosensory cortex, where we have our conscious experience of sensation. If you were to have a stroke in that part of the body, you might lose all awareness of, for example, your hand. The parietal lobe has really specialized specific regions. So there is somewhere in my brain a group of neurons that's totally responsible for my awareness of my right hand. And if I were to receive any kind of damage to those neurons, I would be able to move my hand. I might have like a physiological reaction to pain in my hand, but I wouldn't have any conscious experience of either of those things. That's called agnosia, which means being unaware of something, specifically because of damage to the part of the brain that brings that experience to conscious awareness. So in addition to receiving tactile input in the somatosensory cortex, the parietal cortex is involved in touch and taste, in taste recognition, as well as in bodily awareness. So the parietal cortex is just really important for sensation. And then lastly, the occipital lobe, which is in the back of the brain above the cerebellum, which is a hindbrain structure, is responsible for receiving visual input. So people who have strokes in the occipital lobe have what's known as brain blindness, where their eyes and optic nerves work perfectly well, but because they can't receive and integrate input from those regions in their cortex, they don't have a conscious experience of vision. People with this type of blindness can sometimes have involuntary, like reflexive reactions to things that they see, but they can't articulate having a conscious experience of it. It doesn't reach consciousness because it's not being integrated by the cortex. So the important thing to remember about the cortex is that it is really crucial to our conscious experience of our lives. We could live without it, but we would not be necessarily recognizable as humans because we wouldn't have consciousness. Okay, and this is, again, just for you to study a diagram of the cerebral cortex and the different specific regions we talked about. Okay, we're almost to the end of the neuroanatomy part of today. So there's a ton of neurotransmitters, but not all of them are equally important to psychopathology. So I'm just going to run through some neurotransmitter systems that are implicated in psychological disorders. So one that we talked about last time in the context of psychotropic medication was serotonin, also sometimes known by its chemical name 5-HT. So the serotonin system has been implicated in depression, which is why depression is treated with serotonin reuptake inhibitors, but also in anxiety and obsessive compulsive disorder. Serotonin is also really important in other functions that don't have anything to, I shouldn't say they don't have anything to do with psychopathology, because we'll have a lecture later on psychogastroenterology, which is about the interplay of abnormal psychology and digestion, but serotonin is also really involved in digestion, as well as in sex. So other neurotransmitters that are involved in psychopathology, GABA and glutamate. So these are kind of like the on and off switch of the brain. GABA is the off switch. Whenever it binds to a neurotransmitter, whatever that neurotransmitter does, GABA binding to it causes it to stop doing that thing or to do less of that thing. It's known as an inhibitory neurotransmitter. Glutamate is the opposite. It's an excitatory neurotransmitter. So when glutamate binds to any neuron, it causes that neuron to produce more of its neurotransmitter and to send more of its signals to the next brain region that it communicates with. So GABA in particular is really implicated in anxiety. One risk factor for anxiety could be just either not producing enough GABA endogenously, so not enough of it being made in the brain, or not having enough receptors for it on certain neurons that are involved in the experience of anxiety. So GABA not downregulating the right neurotransmitters to the right degree. GABA is also really implicated in alcohol abuse because ethanol, which is the main ingredient in the alcohol that we drink, is a GABA agonist. It actually binds to the same receptor. So what's happening when you're drinking alcohol is that some of it crosses the blood-brain barrier, so it gets into your brain, and it binds to neurotransmitters in your brain, and it causes them to slow down their rate of firing. So drinking alcohol relieves anxiety for a lot of people. It also affects things like coordination and voluntary movement. It makes your speech slurred because it's binding to motor neurons in your prefrontal cortex and making them less effective at doing their job. Okay, norepinephrine is involved in the stress response, which we're going to talk about in a few slides. So norepinephrine is one of the first steps in the brain of experiencing stress. Norepinephrine is also involved in a lot of resting state functions like attention and just sort of general arousal, not specifically negative stress. We talked about dopamine again last time and a little bit this time in the context of the basal ganglia and voluntary movement. But in addition to just like telling us to move our leg, it also has a more complicated role in motivation. So dopamine is involved in wanting a reinforcer and in enjoying a reinforcer. We'll talk about the difference between wanting and liking in the substance abuse lecture. But basically, dopamine is involved in our experience of unconditioned reinforcement. So when an unconditioned stimulus makes us feel good, that's dopamine. So an unconditioned stimulus that makes us feel good is eating sugar, like eating sweet, highly caloric food. And when we eat something sugary, our brain produces dopamine, and the production of dopamine is part of our conscious experience of enjoyment. Also, when we think about eating something sweet, our brain is also producing dopamine. And so we're not enjoying that sweet thing, but the dopamine is signaling to different parts of our brain that are making us crave the thing and are motivating us to go out and get it. So dopamine is really involved in disorders that involve reward-motivated behavior. So substance abuse disorders, binge eating disorder, and impulse control disorders like gambling. Dopamine dysregulation is also involved in ADHD, which is treated with stimulants based on the theory that ADHD is caused by not enough dopamine in the prefrontal cortex. So not enough activation of the parts of your brain that help you control impulses and help you pay attention. Dopamine is also involved in schizophrenia with the theory that too much dopamine is produced in some parts of the brain, causing parts of the brain that should be quiet to be active and influencing our experience of our sort of internal experiences that aren't tied to sensory experiences in the real world, which can be experienced as hallucinations. And then dopamine is also involved in depression because one symptom of depression is just not enjoying things like you should, not enjoying things as much as you used to. And the dopamine system is also implicated in that. So the last neurotransmitter we're going to talk about today is not actually a neurotransmitter. It's a hormone, but it's involved in neural communication. And cortisol is the downstream product of norepinephrine signaling in the central nervous system. Cortisol signaling in the body is part of our experience of stress. When you're under stress, your body produces excess cortisol. That cortisol flows around in your blood and it creates physiological changes in your body that help to mediate the fight or flight response. So having cortisol in your body is how the sympathetic nervous system tells your heart to beat faster, tells your stomach to stop digesting, tells your airways to relax so that you can get more oxygenated, or constrict rather, so that you can get more oxygenated blood to the parts of your body that it needs to go to faster. And cortisol is really involved in anxiety in the short term, but then if you experience chronic anxiety, the cortisol signaling system becomes dysregulated, and that can contribute to depression. So we'll talk about the cortisol theory of depression and inflammatory stress in a couple lectures from now when we talk about depression. Okay. So neurotransmitters are how our brain cells, our neurons, naturally communicate with each other and how they communicate with structures in our body. When your brain produces neurotransmitters, that's called endogenous neurotransmitter production. Endogenous means occurring within the system, within your body. One tool of psychological medicine is manipulating neurotransmitters with endogenous neurotransmitters, so things that are produced outside your body that interact with the neurotransmitter system. And the two main categories of endogenous neurotransmitters are agonists, which are drugs that mimic the effect of a neurotransmitter, and antagonists, which are drugs that block or inhibit the action of a neurotransmitter. And there are three kind of main ways that psychotropic drugs can act to influence neurotransmission. The most simple way is that a drug can be the same shape and have the same molecular structure as a neurotransmitter so that it binds to the receptor for that neurotransmitter. An example of that is ethanol, which binds to GABA receptors in the brain as an agonist. Sometimes drugs can have a really similar molecular structure that's almost the same, but a little bit different. That allows it to bind competitively. So it binds to the receptor on the neuron. But rather than opening that receptor and causing the neuron to have an action potential, rather than causing the neuron to fire, it blocks the receptor so that endogenous neurotransmitters can't bind. And that actually downregulates the receptor the neurotransmitter, because the receptor is already taken. Endogenous neurotransmitters can't bind to the receptor and cause actual signaling. So agonists that bind to a receptor do so by binding to the receptor and causing it to open, causing the cell to have an action potential. And antagonists work by competitively binding. So binding to the receptor, taking up space, blocking the receptor, and preventing other endogenous neurotransmitters from binding to it. Okay. Next, there are classes of drugs that, rather than working on the receptor of the neuron, work at the other end, at the axon terminal. So after a neuron fires its neurotransmitters, there is basically a certain amount of time during which those neurotransmitters can bind to the next neuron's receptor. And if they don't bind to the next neuron's receptor, the body has to do something to clean them up, to get rid of them, so that we don't have a bunch of like excess neurotransmitters floating around after the stimuli that caused the neuron to fire has passed. That if our bodies didn't do something to get rid of the neurotransmitters, our cognition wouldn't be as efficient. So one way that the body gets rid of excess neurotransmitters is reuptake. So the neuron that fired those neurotransmitters has a mechanism for sucking them back in. And that's called reuptake. So some drugs work by either speeding up reuptake, so causing the neurotransmitters to leave the synapse faster. Sorry, the synapse is just the space between two neurons. So it's the space where neurotransmitters leave one neuron and bind to the next. So drugs that speed up reuptake remove neurotransmitter from the synapse faster and give it less of a chance to bind to the next neuron. Drugs that slow down reuptake do the opposite. They keep the neurotransmitters in the synapse longer, giving them more of an opportunity to bind to the next neuron. And the most common example of that is the selective serotonin reuptake inhibitor, which is one of the main treatments for depression. They work by inhibiting the cell that fired the neurotransmitter, that fired the serotonin, from taking it back. So that makes it available to the next neuron longer. And then lastly, drugs can work by either speeding up or slowing down enzymes that break down neurotransmitters in the synapse. And this is kind of the second way that the body gets rid of excess neurotransmitters. Enzymes are basically just molecules that work with protein, in this case by breaking it down. They break apart the bonds between amino acids and change the structure of proteins. So there's no example on the slide, but an example of a drug that works on enzymes is the Maoi inhibitor. That was an older treatment for depression. It's still sometimes used for people where SSRIs don't seem to be working. And monoamine oxidase is an enzyme that breaks down monoamines, which is the chemical building blocks of serotonin. So whereas selective serotonin reuptake inhibitors prevent the neuron that fired the serotonin from taking it back, monoamine oxidase inhibitors prevent the enzyme monoamine oxidase from breaking down, or sorry, that was confusing. Monoamine oxidase inhibitors prevent the breakdown of serotonin. So they keep the serotonin alive and active in the synapse for longer. Okay, so let's take a break and recognize that that was a lot of information. When we talk about brain regions and neurotransmitter systems throughout this class, we'll do a little mini review every time. So understanding neuroanatomy and how neurons interact and work can deepen your understanding of psychopathology, but having like a really good fluent understanding of this stuff isn't totally necessary for understanding this class. So make sure that you review the textbook. If there's anything that you don't understand, feel free to ask questions on the discussion board in class during office hours. But when you're studying, really focus on the brain regions on this slide and the neurotransmitter systems and drug categories on the last couple slides. Okay. So now we're going to be changing gears a little bit and getting into the actual content of this lecture a little bit more. So in the last lecture, we talked about a lot of unidimensional theories of psychopathology. And this time we're going to be talking about emotions from a multidimensional perspective. So as an example of how many different pathways can lead to kind of the same end result. So just to orient you again, we're going to be thinking about emotions from all of these perspectives, from a biological and physiological perspective, from a cultural and interpersonal perspective, from a developmental perspective, and this is again not on the slide, but from a behavioral reinforcement perspective. And all of these understandings of emotion kind of come together within cognitive behaviorism to try to help clinicians and patients understand abnormal functioning in psychopathology. Okay, so first we're going to talk about how psychologists actually define an emotion. There's three components. One is that emotions are physiological, so they correspond to physiological processes and changes in the body. The second is that they influence and are influenced by cognition. So cognition is our thoughts, and our thoughts tend to be congruent or consistent with our emotions. If I'm feeling the emotion of sadness, my conscious thoughts are more likely to be negative and pessimistic and sad. So that would be an emotion influencing my thoughts. And then lastly, emotions carry with them an action tendency. So emotions evolved to motivate our ancestors to do things that benefited their survival. So for example, when we're feeling the emotion of disgust, the action tendency associated with that is to avoid. If something in the environment makes us feel disgusted, we're naturally, intrinsically motivated in an unconditioned way to avoid it, to not want to touch it, to definitely not want to eat it, to stay far away from it. So emotions carry with them action tendencies. Okay, so first we're going to talk a little bit about the physiology of emotion. So first, we know that specific brain regions can be activated during specific emotional states. The image here is from an fMRI study, which is where it's similar to the MRI we talked about in the last lecture, but whereas an MRI just takes still pictures, an fMRI can actually show brain activation, so brain processes happening in real time. And in the study that this image comes from, they actually had drinkers drink beer while they were in an fMRI machine, and they looked at what parts of the brain were activated by the taste of beer. And the region that's being activated here is in the Mesolimbic System, a region known as the ventral tegmental area, which is one of the downstream targets of dopamine from the basal ganglia. And we know by correlating sort of self-report with activation of that system that when the ventral tegmental area is activated, the person's emotional experience is pleasure. Another example is that when the hypothalamus or the limbic system, the amygdala, is activated, the emotional experience associated with that is fear and anxiety. So in addition to activating specific brain regions, what brain region activation really means is that the neurons in that region are firing. So they're having action potentials. They're sending electrical signals down the axon terminal or to the axon terminal and releasing neurotransmitter. So this lit up area on the fMRI is actually just a lot of neurons in that area being active. So when we feel emotions, those emotional feelings correspond to specific neurotransmitters or hormones in our bodies. And one of the most important examples of that is the emotion of stress and the cascade of neurotransmitters and hormones that are involved in that. So when we feel stressed out, fear and anxiety in the hypothalamus, that region of the brain releases a neurotransmitter called corticotropin releasing factor, which binds to neurotransmitters in the pituitary, which is a region in the brain that has communication with the blood. So the pituitary is kind of the brain's control system for the body. It's where the brain dumps neurotransmitters that get into the bloodstream and communicate really broadly with organs in the body. So I think we'll also talk a lot about it in the context of sleep. But here, when we're having the emotional experience of stress, our brain reacts. It releases CRF to the pituitary, which in turn releases the hormone ACTH, which gets into the blood, circulates widely, eventually reaches the adrenal cortex, which sits right above the kidneys. And the adrenal cortex is where cortisol is produced. And cortisol is kind of the primary stress hormone. It's often used in experiments. So if you intentionally stress someone out in the lab and then take blood from them, like 5 minutes later, their levels of cortisol will be increased. And the increase in cortisol is related to their experience of stress. So a bigger increase of cortisol corresponds to a stronger feeling of stress for the person. So we'll talk about this more again in the depression lecture, and I think a little bit in the anxiety lectures too. But this whole process, the hypothalamus, pituitary, and adrenal communication, is called the HPA axis. And the HPA axis is meant to be an acute stress response. So this is how the body initiates the fight or flight reaction. But because the fight or flight response is designed to be short-lived and temporary, because all of the sympathetic functions are taking away from parasympathetic functions that we really need to survive. So the parasympathetic nervous system is what it controls digestion, getting adequate nutrition, having sex, all of the survival functions. We don't want the sympathetic nervous system to be constantly activated because it would get in the way of our survival if we weren't doing those things. So the HPA axis has an automatic shutoff valve, which is when cortisol gets into the blood, eventually that cortisol crosses the blood-brain barrier and gets back into the brain. And it binds to both to neurons in both the pituitary and the hypothalamus. And when cortisol binds to those neurons, it inhibits their action. So cortisol is an endogenous inhibitor of cells in the hypothalamus that release CRF, or CRH, it's sometimes called, corticotropin releasing hormone. Okay, so yeah, cortisol is kind of like the shutoff valve for the HPA axis. In the body, it activates the sympathetic nervous system, but in the brain, it deactivates the hypothalamus, so it turns itself off. So emotions are biological. Emotions are chemical experiences in our body that we can see in real time by drawing blood or by looking at an fMRI machine. Another aspect of our experience of emotions is that they evolved. So evolutionary biology and evolutionary psychology can help to explain why we have the emotions that we have, and also why some people have maladaptive emotional experiences that are consistent with psychopathology. So one way that we know that emotions evolved is that they're universal. So they're universal to humans, and that's a really important concept in evolutionary psychology that this week's conversation guest, Megan Williams, will talk about. But emotions are also universal to all mammal species. So humans are mammals, but other mammal species like dogs and sheep and rats have emotions that are analogous to human emotions. So when dogs have helicopter tail, which is a specific tail wagging pattern where their tail goes in a circle, that's how dogs express joy and happiness. It's a more specific emotional expression than just tail wagging. It's a kind of tail wagging that indicates that they're really excited and really happy. This image of a sheep is from a research study that designed a coding system to help humans recognize pain in sheep. So the emotion that this sheep is expressing with her face is pain. And the researchers found that you could tell that sheep were in pain by their ears being lowered like this and by their eyes being squeezed shut. And then lastly, the rat, this one is probably the hardest for us to tell, but the emotion that this rat is expressing is happiness. And the best way to tell that with rats is that its ears are relaxed. So if the rat was scared, its ears would be flattened, kind of just like a dog or a cat. And if it was attentive, its ears would be standing up straight. But because it's happy and calm, its ears are sort of somewhere in the middle. They're relaxed. So the function of emotions is that they allow, going back to humans, they allow us to communicate with other humans. And because socialization and living in a social structure has been important to human survival, emotions have been evolutionarily conserved for humans. But they serve a similar function for animals too. They are a means of nonverbal communication. So again, emotions are universal. And the work of a psychologist, Paul Ekman, has identified 6, possibly 7, but the jury's still out on one of them. But 6 emotions that are totally universal to all human societies, all cultures, including hunter-gatherer cultures that have had limited contact with other human populations, which again is a concept that almost Dr. Williams, she's a PhD candidate, but she'll be a doctor soon, talks about in the study guide for this week. So the 6 basic human emotions are anger, disgust, fear, happiness, sadness, and surprise. And the faces on the right of the screen are expressing each of those emotions. So you can probably tell just by looking at them which emotion that person is expressing. And Paul Ekman did studies where he went to really remote hunter-gatherer tribes and showed them pictures of people making these expressions. And despite not interacting with outside cultures, these people were still able to recognize the emotions that were being expressed in the picture. And they were able to produce those emotions themselves. They said that they basically have the same emotions and they express them in the same way. So a principle of evolutionary psychology, again, is universality. If something is universal to a species, it means that it existed in a common ancestor of that species. It likely didn't spontaneously evolve in multiple human populations. It's likely evolutionarily very old. And if a behavior is evolutionarily conserved, we can infer that it's associated with survival, that it promotes survival. But Megan will be talking about other types of evolutionarily conserved behaviors and phenotypes that don't necessarily confer survival. So make sure that you pay attention to that distinction when you're listening to the study guide this week. So in addition to being evolutionarily conserved and therefore probably serving an adaptive function, a survival function, we can actually, to some extent, pinpoint the origins of individual people's emotional experience in their genes. So we're going to talk to Dr. Eisen, who's a USA faculty member who studies behavioral genetics, next week in the study guide for one of next week's lectures. But just a very quick introduction to genetics. A gene is a piece of information on your DNA, so on one of your chromosomes, that codes for proteins. And proteins are basically the building blocks of the human body, including they're the building blocks for neurotransmitters, for receptors on neurons and for the neurons themselves. So genes tell your body what proteins to produce, and then those proteins go on to influence the structure of your brain and its function in terms of neurotransmitter signaling and receptors. So two important pieces of terminology. A genotype is just the set of genes that you have. So it's something that can be read out on a computer. It's just a series of base pairs, like a code. Phenotype is the observable expression of your genotype. So if you have genes that make you vulnerable to anxiety, that's your genotype. But if you feel a lot of anxiety and engage in a lot of anxious avoidant behavior, that's your phenotype. Maybe more specific example of that would be if you have genes, if you have 2 recessive genes for blue eyes, that's your genotype. The eye color itself is your phenotype. It's the observable manifestation of your genes. So we're going to talk in the next couple slides about a couple different, genotypes and phenotypes that are associated with psychopathology. But first, genes are kind of the currency of evolution. So when we talk about traits or behaviors being passed down or evolutionarily conserved, what we're actually talking about is specific genes that encode for specific proteins being passed down throughout evolutionary history. So the way that genes get passed down is that, again, all of us have 23 pairs of chromosomes. When we reproduce, so when we either produce an egg cell or a sperm cell, that cell contains 50% of our genes. Basically, each chromosome has 2 sides to it. And when we produce an egg cell or a sperm cell, each of those chromosomes gets split and half goes into that cell. So every time we reproduce, we have a 50% chance of giving a specific gene to our child, and the child's other parent can also give them a gene. So not every gene that we have in our body is passed down to all of our descendants. There's a 50% chance for each gene that we have. So organisms with certain behaviors or certain physical appearance, like aspects of their physical appearance that makes them more desirable to mates, it gives them more chances to reproduce, or it just helps them survive, or it helps their offspring survive. So it helps them protect their offspring and nurture them and promote their offspring survival. Those organisms are just more likely than other organisms to pass down their genes because they're more likely to survive long enough to reproduce. They're more likely to get a mate, and they're more likely to keep their kids alive long enough for their kids to reproduce. So the genes that are linked to those phenotypes are just more likely to be present in the next generation because the people that have those genes are the people who are producing the next generation. So the book is going to talk a lot about the diathesis stress model, which is basically just a reminder that even if genes can explain a lot, they definitely can't explain everything about human behavior. And the diathesis stress model is one example of how genes interact with our environment to produce risk or protect us from psychopathology. So the diathesis stress model is basically that diathesis is risk that we're born with. So often it's talked about as genetic risk. So a genotype that is related to a phenotype that is pathological. In this example, the genetic vulnerability is to alcoholism. And we know that because the dopamine system is involved in alcoholism, this could be genes related to dopamine production and signaling. The stress part of the diathesis stress model is life events. They don't even have to be negative life events. So in this example, the stress is just exposure to alcohol. If someone has a genetic vulnerability to alcoholism, but just happen to never drink alcohol, maybe they live in a culture like they're Muslim or Mormon and just never have the opportunity to try alcohol, that person is really unlikely to become an alcoholic. But if that person was in an environment where they drank alcohol, even in a way that initially wasn't pathological and wasn't any different from peers who drink alcohol, because of their genetic vulnerability, that exposure to alcohol could be enough to produce substance use disorder. So in the example here, these are 2 people who differ in diathesis but have the same stress. So maybe like 2 frat brothers who do the same amount of binge drinking in college. One of them has a genetic predisposition to alcohol and the other one doesn't. So the same amount of binge drinking in college is enough to cause one of these 2 frat brothers to go on to become an alcoholic, but not enough for the other one because that second person, person number 2, doesn't have enough risk genes for alcoholism. So the diathesis stress model is just kind of an important reminder that although we talk about evolution and genes, and we talk about how these things are associated with risk for psychopathology, genes alone are not enough to reduce psychopathology in most cases. So an example of the diathesis stress model is the short allele of the serotonin transporter gene. So the serotonin transporter gene basically encodes for proteins that are involved in the movement of serotonin in the synapse. So it has to do with how much serotonin is available in the body, similar to SSRIs. And there are 3 different genotypes that encode for this same protein. And again, all of us get one gene. We have redundant genes. We have 2 copies of every gene. We get one from our mother and one from our father. The serotonin transporter gene has 2 varieties, the long allele and the short allele. And an allele is just a variant on a gene. So a gene that has more than one manifestation, like more than one type, is known as an allele. So the serotonin transporter alleles are known as alleles because there's 2 versions. One is short and one is long. That's just the length of the code that encodes the protein for that gene. So people with 2 copies of the short allele are known to be at elevated risk for psychopathology. And people with 2 copies of the long allele are actually protected against psychopathology. But this genotype alone is not enough to predict risk for psychopathology. It has to interact with life adversity. So people with 2 copies of the short allele who are exposed to difficult life experiences in childhood, it could be something chronic like child abuse or child neglect, having a parent with a substance use disorder, or it could be a shorter term acute stressor like being involved in a school shooting. But without those environmental stressors, having 2 short serotonin transporter alleles is not enough to usually to tip someone over into having psychopathology. And it's also important to remember, and again, we'll talk about this with Dr. Eisen next week, but childhood adversity itself is not separable from genes. We get our genes from our parents, and our parents create our childhood environment unless we're adopted. So if your parent happens to have 2 short versions of the serotonin transporter allele, and they had a stressful, difficult childhood, maybe that genotype led to an alcoholic phenotype for them. So maybe they have a substance abuse problem. And having a parent with a substance abuse problem is a form of childhood adversity. So if the parent then passed on their short copy of the serotonin transporter gene to their child, they would be putting their child at both genetic and environmental risk for growing up to develop psychopathology of their own. So this is a very famous study exploring the interaction between the serotonin transporter allele, childhood adversity, and the outcome of, in this case, depression. So what we're looking at here, the red line, are people with 2 copies of the short allele, so the short genotype. The bottom line here in purple are people with 2 copies of the long allele. And in the middle are people with 1 copy of the short allele and 1 copy of the long allele. And this is the most common phenotype because if you have the short allele, you have to get 1 short allele from your mom and 1 from your dad. And same with the long allele. So you could have 1 parent with the long genotype and 1 parent with the short genotype. You'd be guaranteed to have the short long genotype. So this is a study of a large group of people who varied in their genotype and also in the extent to which they experienced maltreatment as children. So first, we're looking here at children who didn't experience any maltreatment, all the way over on the left. These are people who had very low levels of stress in their early environment. And we can see that regardless of which genotype they have, so regardless of their diathesis, their risk for psychopathology, in the absence of childhood maltreatment, there are no differences in depression. People with the high-risk genotype, the short allele, don't have any higher rates of depression than people with the low risk genotype, the long allele. In the middle here are people who experienced what the study is calling probable maltreatment, but basically some degree of difficulty in their childhood, but no overt or known child abuse. So these are people who had stressful, difficult childhoods. And here we start to see separation where people with at least one copy of the short allele, so short long and short, people start to have higher risk of depression than people with two copies of the long allele. And those group differences get even bigger when people experience severe maltreatment in their childhood. But one of the most interesting things to look at is that people with the long allele actually, regardless of how much maltreatment they experience, don't have higher risk of depression. So whereas the short allele is a high risk gene for depression, the short genotype, the long genotype is actually a protective genotype against depression. So another example of a high risk genotype that alone, in the absence of environmental stressors, doesn't necessarily have an impact on psychopathology, is what's known as the warrior enzyme. So we talked about how enzymes break down neurotransmitters in the synapse, and that affects how available those neurotransmitters are and for how long. So there are alleles, so different versions of the gene that encodes for what's known as the Compt enzyme. And I actually don't remember what Compt stands for, but it's a very long word. Basically, what this enzyme does is it breaks down neurotransmitters in the prefrontal cortex. So again, that's the part of the brain that's responsible for thinking ahead, for planning, for rational thinking, for controlling our impulses, for regulating our emotions. And it turns out that when people have fewer of these enzymes, when their body produces less of the enzyme that breaks down neurotransmitters in the prefrontal cortex, under low stress conditions, so most of the time, those people actually show some advantages. So they do better in school, they do better on standardized tests, they're better at logical thinking, they're better at planning, they have better memories. But under stressful conditions, their sort of thinking ability breaks down. Their prefrontal cortex becomes less efficient because the slow breakdown of enzymes makes their prefrontal cortex function less efficiently under conditions of stress. So under conditions of repeated, frequent, high stakes input, breaking down neurotransmitters slowly is actually a disadvantage, whereas under normal conditions, it's an advantage. So people with fast breakdown have what's known as the warrior with an A genotype. So these are people who show really good performance under stress, and they tend to be really brave. But they, under calm, sort of normal conditions, they don't necessarily perform as well at complex thinking tasks. So the pictures here on the left are kind of illustrating what roles in early human societies people with these different genotypes might have had. And it's starting to hint at a discussion question for today's lecture, which is basically, if some genotypes confer risk for psychopathology, even if that risk is only visible under high stress conditions, why would those genes be preserved in human populations? Why would people who have high risk genes for anxiety have any reproductive success or have more reproductive success than people with genes that protected against these phenotypes? So getting back to gene environment correlation, which we talked about in terms of childhood adversity with the serotonin transporter allele, with the Compt enzyme, people who have this enzyme, who produce lower levels of this enzyme, so people with the warrior with an O genotype, because we're conscious beings and have a self-image, like a sense of who we are as people, people with this genotype know that they don't perform as well under stress. And so they're motivated to avoid high stress situations and seek out situations where they thrive and succeed. But over time, this intentional behavior, this human conscious reaction to one's own genotype, can actually have an influence on the behavior. So the behavior is genetically predisposed, but then our conscious choices and the environment that we select ourselves into has a further influence on our behavior. So if I'm someone who has the warrior genotype, maybe I recognize that I do well in academic settings, I am a good test taker, I'm not good under pressure, so I wouldn't go into like a high stakes job like being a trauma surgeon or a firefighter, because I know that under conditions like that, I tend to get really stressed out and really anxious and not think very clearly. If I avoid high stress situations to a really high degree, I might grow up to be someone who doesn't have any strategies for coping with stress, and I might develop an image of myself as someone who can't handle stress. And because stress is kind of inevitable, even in the academic settings that I might select myself into, I might over time, because I never learned strategies to handle stress, start to break down under normal stress conditions like exams in college. So this is one way that maladaptive anxiety can come about. Someone with this high-risk genotype creating an environment for themselves that doesn't expose them to enough stress, doesn't let them learn strategies for coping with it. Okay, so we've already started talking about ways that the environment can influence emotions by interacting with our genotypes, but separately from different risk levels in our genes. So separately from serotonin transporters or prefrontal cortex neurotransmitter breakdown, regardless of our genotype, the environment can influence our emotions. And some environments are just stressful. So an example, again, is childhood adversity, growing up with a parent who has a substance use disorder, or growing up with a parent who's abusive. Constant stress causes constant activation of the HPA axis. So the hypothalamus constantly produces CRH, which constantly causes the pituitary to produce ACTH, which constantly causes the adrenal glands to produce cortisol. And even though the cortisol is in our blood shutting down the whole cycle, if our environment continues to be stressful, the cycle will just start over and over again. So we'll constantly be in this loop of producing cortisol in our blood, fight or flight reaction. Over time, if we're exposed to enough constant high stress, this can sensitize the stress reaction. So it can actually make it more likely that we'll have a stress reaction even under lower stress conditions, because our brain is just so used to reacting to stress by producing CRH, and our body is used to reacting to stress by producing cortisol. And in addition to that, chronically high cortisol over time can lead to depression by causing dysregulation of the HPA axis. Over time, if cortisol is constantly in our blood, it can cause a breakdown of the negative feedback part of the HPA axis. So eventually, cortisol stops having as strong an effect on the hypothalamus, and it stops being able to downregulate CRH production. So that leads to just runaway high cortisol levels called hypercortisolemia, which is one way in which stress, regardless of our genotype, regardless of our risk factors for experiencing anxiety under stress, enough stress can produce psychopathology. So part of the stress diathesis model is that there may be some genotypes that, regardless of their environment, will develop psychopathology. And there are some environments that can produce psychopathology almost regardless of our genotype. So moving on to another piece of this integrative approach to emotions, we also have to think about culture and human society in both the experience of emotions and the expression of emotions. So although 6 emotional responses are universal to all humans and most animals, culture and upbringing for humans, at least, because as far as we know, humans are the only animal that has a culture, culture can influence or determine what emotions we experience. So which situations trigger which emotions. So one example of this is disgust. Before like 1970, Americans did not eat sushi at all. So if you talk to someone in like 1960 and told them that you really enjoy a dish that's made from seaweed, rice, raw fish, and vinegar, they would be disgusted by that because their culture hadn't exposed them to that enough that they recognized it as food, and they hadn't had any chances to experience it and learn that it's actually delicious. So pre-introduction of sushi to America, this image would evoke disgust, and nowadays for most of us it evokes craving or pleasure, or it's at least neutral. in the 1850s, this image of a woman wearing pants, for some people would trigger anger, whereas now women wearing pants is totally common, totally normative, totally acceptable in our culture. So this image doesn't cause us to have that emotional reaction anymore. In the 1850s through like the early 1900s, women wearing pants was a really highly politicized issue. So it caused anger for some people. And that really was influenced by culture. There's nothing inherently negative or stimulating about pants. Our culture sort of decides for us the reaction that we'll have to them. A more recent example would be this image. So say that you're like on the subway or standing in line at the grocery store and someone behind you starts coughing. Last November, you probably wouldn't have had a super strong reaction to that. You might be like a little concerned, not wanting to catch a cold, a little annoyed, maybe a little bit grossed out. Nowadays, someone coughing on you might be enough to produce the emotion of fear. And that's because of our current cultural context. So there's nothing necessarily that's changed about our society and its approach to illness, but there's a specific illness going around that is dangerous. So our cultural context, our period in time, influences the emotion that we feel in response to this stimulus. Again, there's nothing inherently scary about coughing. And then lastly, culture can influence not just the emotions that we experience, but also the way that we choose to express emotions. So this is an image from a jazz funeral in New Orleans. So in most cultures, when we attend funerals, the sort of appropriate emotion to express is sadness. But in the New Orleans jazz funeral culture, or like in some, like in Irish wakes, it's actually more appropriate to express joy and appreciation at a funeral because it's a celebration of life. So that isn't to say that people who celebrate funerals this way don't feel sad, but it's an example of how culture can influence our choice to express emotions, which emotions we share with other people. And the choice to express or suppress emotion, which is heavily influenced by culture, can influence psychopathology. So that is one social influence on emotion. The way that our culture teaches us and encourages us to express or suppress emotions. Another example of social influences on emotion is relationships. So being in social relationships really helps us to regulate and manage our emotions. Loneliness itself isn't a form of psychopathology, but we know that loneliness is a huge risk factor for psychopathology. And loneliness, in part, acts on producing psychopathology by not giving us enough outlets to manage our emotions. Humans are social animals, and our social relationships help us process our emotions in healthy ways. So taking a human out of their normal social context and isolating them can cause the development of psychopathology in someone who maybe doesn't have genetic risk and who doesn't have any other environmental stressors. And then again, social roles. So this is more about the way that we feel that we have to express or suppress our emotions. There are some aspects of culture that stigmatize emotional expression. So, for example, one stigmatized emotional expression might be like happiness at a funeral, unless it's a jazz funeral or a wake. An example that's more closely related to psychopathology is gender in the United States. So we know that there are sort of gender norms about how much emotion people are sort of allowed to express. And in our culture, women are allowed, encouraged, and sort of accepted when they express emotion more than men are. And we know that this has an impact on psychopathology. Women are more likely to be diagnosed with an anxiety disorder or with depression. We don't know for sure if women are more likely to have those disorders, though, because our ability to diagnose depends on a person disclosing. We'll cover this in the next lecture, but we don't really have a lot of totally objective biological ways to diagnose psychopathology. There's a lot of reliance on self-report. So if society influences a person's comfort level with expressing emotion and sharing it with a mental health provider, that can affect the extent to which someone is diagnosed with an emotional disorder like anxiety or depression. It can also affect the extent to which the person themselves recognizes that their emotions are problematic and seeks help for them. So one theory of why men have lower rates of diagnosed anxiety and depression, but higher rates of diagnosed substance abuse, is that because of gender roles that prevent men from expressing negative emotions, they are more likely to self-medicate with substances and develop substance abuse disorders. Whereas women may be more encouraged to engage in behaviors that can promote the development of anxiety disorders, such as avoidance. Women might be more socially excused for saying like, oh, I'm sorry, I can't do that thing, I'm scared of it. That might be stigmatized in a man. So a man might feel like he has to do the thing that he's scared of, but cope with it in a maladaptive way by, for example, getting drunk first. Whereas a woman who's coping with that anxiety in a maladaptive way might just avoid it and experience less social stigma for doing that. Another example that we'll talk about in the personality disorder lecture is this idea that borderline personality disorder and antisocial personality disorder are two sides of the same coin, where their influence is really, or their expression is really influenced by gender roles. It's possible that those two disorders have many shared genetic risk factors, and gender and socialization and the environment that boys and girls are exposed to is what influences the different expression of psychopathology. So culture influences emotional experience and emotional expression, but getting back into a behaviorist model, we definitely know that emotional responses can be learned and learned through classical conditioning. So like we talked about last time, unconditioned stimuli are things that are inherently positive or negative. One example is food. And we know that the taste of food activates neurons in the ventral tegmental area. And when those neurons are activated, we feel pleasure. We don't have to learn that association. However, if you're someone who enjoys, like, say, McDonald's or Starbucks, you can develop a conditioned association between the pleasure that you get from eating the food there and just the experience of going there, the symbols that represent those stores. So like the McDonald's logo, the Starbucks logo. If you develop a conditioned association for that logo, seeing a McDonald's sign on the highway can also activate those neurons in your ventral tegmental area and also create motivation and craving and a sense of pleasure, just the same way that the food naturally does. So that's an example of classical conditioning of an appetitive behavior, seeking out and enjoying food. We talked last lecture about Freud's case study of little Hans, which was an example of classical conditioning of fear. He had a negative experience with a horse. The horse was neutral before that negative experience, but because his reaction to seeing something die was unconditioned and negative, that reaction got paired with the stimulus of a horse, and he developed a fear of horses. So in the future, horses caused the same emotional reaction for Hans that death had originally caused for him. Another example that we're all probably familiar with is video games and phone games. Over time, these games use symbols like the Candy Crush symbols or sounds or vibrations. Those sounds and vibrations are paired with rewards. So like leveling up, there's a specific sound tied to that. Over time, those sounds can create activation in the ventral tegmental area and cause our bodies to produce more dopamine. And we become reinforced for engaging with those things, even though we don't always get rewarded for doing it. And if you look at the further readings for this lecture, there's actually an article about that, about how video games kind of train your brain to almost become addicted. Okay, so we're getting to the end. I'm going to take a quick water break because this is a very busy slide. Okay. So I started talking about this when I was talking about gender roles and psychopathology, but our behaviors, our intentional choices can have an influence on the development of psychopathology. So over time, the choices that we make, the behaviors that we do can influence our environments, they can interact with our genes, and that can lead to the development of new emotional responses or the strengthening of existing emotional responses. And in some cases, that leads to psychopathology. So one example of this is substance use. Alcohol, as an example, is positively reinforcing for most people. Most people find drinking enjoyable. It's also negatively reinforcing. So negative reinforcement happens when we engage in a behavior to escape from a punishment, to escape from something negative. And in the case of alcohol, people drink to escape from negative feelings like anxiety or sadness. So drinking is enjoyable. It also helps us avoid unenjoyable emotions. Drinking is really reinforcing. For people who are more sensitive to that reinforcement, so a genetic risk, a diathesis, or for people who have a lot of negative emotions, which can also be diathesis, or it can be just their environment is really stressful and really difficult. Drinking can be more reinforcing, and that can set the stage for the development of substance abuse. Now, on the other hand, when the body becomes dependent on substances like alcohol, not drinking, so not adding all of those exogenous GABA agonists to your system, produces anxiety. So when you become physically dependent on alcohol, it influences the way that your brain produces neurotransmitters, and the absence of alcohol can cause anxiety. So that's an example of learned behavior directly influencing your emotional experience. Another example that we'll talk about a lot in the anxiety lectures is avoidance. So avoidance is a behavior that is negatively reinforced. Anytime we do something that helps us escape from anxiety or depression or sadness, it reinforces that behavior because those emotions are negative and we don't like feeling them. So with anxiety, if you avoid situations that make you anxious, it's a great way to relieve anxiety in the short term. But in the long term, it actually can make those situations even more anxiety provoking because, like we talked about with the warrior enzyme, if you never give yourself the chance to learn to cope with anxiety, to develop strategies to deal with it, or to learn that the things that cause you anxiety really aren't that dangerous, like little Hans with the horses, that can maintain anxiety and over time make it worse. Another example of the interplay of emotions and intentional behaviors is learned helplessness. So the textbook talks about this. But this was a really terrible experiment that was actually done at my college, where the researchers put dogs in cages and exposed them to uncontrollable shocks. So the dogs were just in these metal cages, the floor was electrified, and sometimes the researchers would just shock them. It was painful and aversive for the dogs, and they were given no way to escape it. So over time, the dogs that were exposed to these uncontrollable shocks, they learned that nothing they did would make it stop. They didn't learn that right away. They tried to escape. They scratched on the bars. They threw themselves against the walls. They tried to keep their feet off the bottom of the cage to avoid the shocks. But over time, they learned that that didn't work. That didn't help them avoid the shocks. So over time, they stopped trying. And then the key part of this experiment is that the researchers gave them a chance to escape. So after many, many, many shocks in the cage where they couldn't escape, the researchers actually opened the door and started shocking the dogs. And the dogs that had been exposed to uncontrollable shocks didn't try to escape even when they had a chance to. So the researchers considered this sort of an animal model of human depression. One way that depression can develop is when you have a ton of childhood adversity, lots and lots of negative experiences in your childhood, and no way to control it or make it better for yourself. Over time, you might develop a coping strategy that is apathetic, that conserves energy and resources by not trying to escape anymore because you've learned that your behavior doesn't exert any control over your environment and it's just a waste of resources to try. So that's an example of how environmental stress can influence behavior and also influence the emotions that you experience in response to that stress in what is probably a self-protective way. Apathy in the face of uncontrollable stress might be more adaptive, a better way to conserve your energy and resources than continued efforts to escape. So we're getting towards the end. We're getting to higher order stuff, to conscious thoughts. We know that although emotions are encoded in our genes, they're preserved by evolution, they're influenced by culture, they're influenced by learned conditioned behaviors. They're also influenced by our conscious thoughts, which means that we can actually exert some control over our emotions with our conscious brains. But one example of how conscious thoughts can influence emotions is actually cognitive biases. So one sort of thinking error that people make is misattributing arousal. So some physiological responses are not very specific. Specifically, the response to fear and the response to excitement have some things in common. They both involve elevated heart rate. They both might raise your blood pressure. They both might cause you to breathe faster. So it's possible to experience that physiological sensation and not know whether it's being caused by stress and danger or excitement. And in some cases, people sort of make mistakes about what's causing it. So a famous example of this was a study in 1979 where researchers had attractive men or women stand in the middle of a really high, scary suspension bridge and ask out people who were walking past them. So they would like approach someone on this bridge, introduce themselves, and ask for their phone number and ask them to go on a date. And then the researchers measured how many people said yes to giving their phone number and also how many people later called the person whose phone number they got. And what they found was that when volunteers were standing on a really high, scary bridge and approached people asking for a date, they got way more phone numbers and way more callbacks than when those same volunteers, so the same level of attractiveness, approached people before they got on the bridge, when they were still on solid ground. And the conclusion that the researchers drew was that people on the bridge were experiencing arousal because of fear, because they were in a really high place and they were feeling a sense of danger. But they misattributed that arousal to excitement and sexual interest. And they thought, mistakenly, that they were more attracted to these volunteers than they actually were, and more attracted to them than they would have been had they been in a less arousing situation. Another example of research that manipulated arousal and manipulated the environment to influence how people interpreted that arousal was a study that was done with people who were asked to wait in a waiting room before an experiment started. They were given what they were told was a study drug, a drug that would I think they were told that it was a drug that would help them concentrate, or it would help them do something that was kind of separate from physiological arousal, something that didn't prime them to think about physiological arousal. So they were told that they needed to take this drug and then sit in a waiting room and wait 10 minutes for the drug to kick in before they started the study. But what the participants didn't know was that the drug actually didn't make them concentrate better. It actually made their heart race and it made them breathe faster, and it basically activated their sympathetic nervous system. So they're sitting in this waiting room, they're experiencing physiological arousal in the absence of any external stimulus to cause it. It was caused by an exogenous drug that they took. So what the researchers did was they manipulated what the waiting room was like. After the person took the drug and started feeling the physiological effects kick in, they had a confederate come in. And a confederate is just like the person asking for dates on the bridge. It's someone who's helping the researchers. They had a confederate come in and either act really, really angry. They were yelling at the researchers for making them wait a super long time to do the study, or really excited. They were just sort of randomly bouncing around and wringing their hands, acting super happy and excited. So the study subjects who had taken this drug that created physiological arousal were sitting in the waiting room looking at this other person, either expressing a lot of anger or a lot of excitement. And then eventually they were taken back by the researchers and asked to fill out questionnaires about their mood. And the people who had been in the waiting room with the angry person were more likely to report that they felt angry, whereas the people who were in the waiting room with the happy person were more likely to report that they felt happy. And the key to this is that another group of subjects, a control group, were given a drug that didn't cause physiological arousal. They were given a placebo, and we'll talk about that concept in the next lecture. But basically, they were not given any substance that would influence their physiological arousal, so they weren't actually experiencing any heightened physiological arousal. And for those people, the emotions that the confederate was expressing didn't really have any impact on their mood. So what the researchers inferred from that study was that when you're experiencing arousal, you look to cues in your environment to help you figure out what emotion is causing that arousal. Sometimes figuring out your emotions is a conscious cognitive process. So although emotions are physiological, our sort of higher order reasoning and intelligence can help determine how we experience that emotion, which emotion we experience. And one of the further readings is an article about how reality shows, and specifically reality dating shows, take advantage of misattribution of arousal to kind of create these fake artificial TV relationships. So feel free to read that article and talk about it on the discussion board for this week. Okay, so we're getting really close to the end. This is one of the last slides, and this is kind of bringing it all home, talking about how cognitive behavioral therapy works to influence people's experience of emotion and therefore their symptoms of psychopathology. So one of the key principles of cognitive behavioral therapy is that we can influence our emotions through conscious thought. We can control our emotions to some extent. We are the authors of our experience, and we can decide how we choose to interpret the physiological sensations that we have and the experiences in our environment. So an example of the cognitive model is how a person responds to a failure. So say that someone fails a test that was really important to them, or they rush a sorority and they don't get in. It's totally natural and normal to feel sad and bummed out about that. When you really want something and you don't get it, that is an unconditioned stimulus. It naturally makes you feel bad. You don't have to learn that. However, there are individual differences in the way that we interpret that feeling of sadness and the mood congruent thoughts that we have. So way back in the beginning of the lecture, I said that part of emotion is cognition, is having thoughts that are consistent with the nature of the emotion. So for some people, being sad can lead to really negative, pessimistic thoughts like, I suck, I'm really bad at this, I'm never gonna make friends, nobody's gonna like me, I'm not gonna get into any sororities, I'm gonna fail all my classes, I'm not gonna get to go to grad school. So this sadness and disappointment leads to really excessively negative thoughts. And why this happens, it can be because of environment, it could be because our parents modeled this kind of thinking for us. It could be because we grew up in an uncontrollably stressful environment and we're just used to things not going well for us. It could be because we have a genotype that puts us at risk for not being able to handle stress and failure well. This is one thing that can happen to people with the warrior, with an O, genotype under stressful conditions. They can kind of break down, get stuck in their heads, and have trouble thinking through the situation to find good solutions. So there can be, and also social roles might influence how you take a failure and how you interpret it. So there's all kinds of multidimensional influences on how we react to our emotions and the type of thoughts that we have. But the CBT model suggests that the proximal thing, the thing that we have the most control over and the thing that therapists can intervene on, is the type of thoughts that someone focuses on when they're having an emotion. So people with depression are way more likely to have these big negative thoughts about a failure. Thoughts like, I'm always going to fail at this, I'm bad at this, I'm not a good person, people don't like me. Really general, global, very permanent interpretations of the failure. A cognitive behavioral therapist might teach someone to challenge those thoughts and actually try to reframe them, rather than interpreting every failure as a sign that you're going to fail in the future. Focus on things that you can control to keep that failure from happening again. So if you failed a test, you could think, well, I'll study more next time. I'll ask for help. I'll go to the learning center. I will set aside more time to study. I'll try to sleep more the night before. So coming up with like concrete strategies to do better rather than focusing on the failure and letting your thoughts remain congruent with the emotion that you feel because of the failure. People who think that way, either sort of naturally because of their genotype or their environment growing up or their social roles, are much less likely to develop depression. But people can learn to think that way. So people who have depression can train themselves to replace their automatic negative thoughts with these more positive, adaptive, helpful thoughts. And that is one way that people can recover from depression. So this is a way of reacting to our present thoughts, but it's also a way of controlling our future thoughts and our future emotions. Because if you react to failing a test by saying, I suck, I'm always going to be bad at this, you don't have any motivation to try to do better next time. If you react by saying, there are things about this that I could control, I could study more, I could ask for more help, you're more likely to seek out those things that will make you do better, and you're less likely to fail in the future. So by changing your thoughts in the present, you're setting the stage for having different emotions in the future. You're making it less likely that you'll fail and making it less likely that you'll feel that type of sadness again. So the cognitive behavioral model suggests that all of these multiple things influence our emotions, but the proximal thing that influences our emotions is how we interpret them. It's our conscious minds, it's our way of thinking. And by changing the way that we think through active practice, we can somewhat change the emotions that we're having in the present moment, but we can have a lot of control over the emotions that we feel in the future. Okay, I'm gonna take a quick drink. So, in summary, emotions are biological. They are the product of neurotransmitters in our body. They evolved. So we have them because at some point they were adaptive to our ancestors. There are 6 basic human emotions, and to varying degrees, those emotions are all implicated in psychopathology. So even though emotions are adaptive and necessary and universal, sometimes they can go wrong and lead to psychopathology. Our experience and expression of emotions is influenced by our culture. So our culture tells us sometimes how to feel about ambiguous, neutral, conditioned stimuli. Things like sushi and women wearing pants and someone not covering their mouth when they cough. Our reaction to all of those things is very culturally dependent. Culture and social roles also influences the way that we express emotion. So how much we feel like we can express emotion, how much social support we can seek out and express emotion to other people, and also which emotions we think are appropriate to express and which ones are not appropriate to express in given situations. So social roles can influence how much we try to suppress our emotions and how much we seek out support for our emotions. And those things in turn can influence psychopathology. So emotions are learned. Not all emotions are inherent biological reactions to stimuli in the environment. Some emotions are conditioned. And some behaviors that are conditioned, that are reinforced by the environment, that are in some ways reinforced by enhancing or escaping from our emotional experience, can actually set the stage for more emotional experience in the future. So an example of an intentional behavior that influences emotion is the choice to drink alcohol. That's a conscious behavior, but the part of it that's not conscious is the natural reinforcement that we get from drinking alcohol. And further downstream of that, what's also not conscious and not within our control is the effect of physiological dependence on alcohol. And both of those things influence our emotional experience. Another example is avoidance. So it's understandable to want to avoid situations that make you anxious, but people who avoid excessively or who avoid too many situations or people who avoid too much can go on to develop anxiety disorders because they don't learn strategies to regulate their anxiety while they're having life experiences. And then lastly, emotions are influenced by and influence our cognition. So we can exert control over our emotions by actively controlling the way that we think and the way that we interpret those emotional reactions. Oh boy. Okay, so we spent this whole lecture talking about emotions because emotions are key to psychopathology. Most of the disorders that we will talk about in this class are either characterized by dysregulated emotions, so having emotions that are too strong or having emotions that are inappropriate to the situation, or by problematic behavioral responses to emotions. So by avoiding anxiety, avoiding anxiety, anger, and sadness with substance use, or by having a cognitive tendency to negatively interpret emotions in ways that influence future behavior in ways that are maladaptive. And this was a very brief summary of all of that. We'll go into all of these processes when we talk about specific disorders later in the semester. So the key to the cognitive behavioral model, which is the explanatory model that we'll use to talk about the development, the maintenance, and sometimes the treatment of all of these disorders, is that we can treat psychopathology by helping people to understand all of the influences on their emotions, but also helping them to understand that they exert choice and control over their emotions. You can't not feel your feelings, but you can choose how you react to your feelings, and you can choose how you interpret your feelings. And those choices can have a big impact on the frequency and intensity of emotions in the future. So one example of this that we'll talk about a few lectures from now is panic disorder. So some people, panic attacks are really common, and they're often triggered by physiological arousal. So one way that you can interpret physiological arousal is in a way that makes you feel anxious. And there are just some people who, because of their genes or because of their early environment or because of their recent experiences, are more likely to feel anxious in the presence of physiological arousal. And because anxiety influences the types of thoughts we have, because our thoughts are congruent with our emotions, if someone is having this physiological arousal, feeling breathless, feeling that like their heart is racing, and they feel anxious, a thought that's congruent with anxiety is, oh my God, I'm having a heart attack, I'm gonna die. And of course, if you have that thought, it contributes to more anxiety. Because before you were experiencing physiological arousal, but now you've interpreted it as something really dangerous. And being in danger activates your sympathetic nervous system even more. So now you're having cortisol flowing through your blood, you're experiencing the fight or flight reaction, the racing heart and breathlessness you were experiencing before are even more intense. And that kind of validates the thought you were having. Like, the sensations that made me think that I'm having a heart attack are still here and they're even stronger. I must be right, I'm definitely having a heart attack. That cycle eventually can develop into a panic attack. So it starts with physiological arousal. That physiological arousal is interpreted as anxiety. That anxiety influences our cognitions, which in turn further influence our emotions. And that can lead to uncontrollable, dysregulated emotion, and in this case, a panic attack. What someone can learn in cognitive behavioral therapy is to recognize that cycle, to understand that that's what's happening in their body when they have a panic attack, and to sort of see objectively, like a scientist, all of the parts of that cycle so that they can make a choice to act on the cycle and have a different response. So someone with panic attacks who goes to therapy and learns these skills can learn alternate thoughts that they can intentionally remind themselves of when they're having this physiological activation and the anxiety that comes with it. They can tell themselves, now wait a second, this has happened to me so many times before. I've thought that I was having a heart attack and it turned out to be nothing but a panic attack. So what's more likely to be happening is that I'm just out of breath because I was running or because I took the stairs instead of the elevator. There's a benign explanation for this physiological reaction I'm having, an explanation that doesn't produce further anxiety. And over time, learning to kind of focus on that explanation, learning to break the cycle of anxious thoughts, anxiety because of the thoughts, further anxious thoughts because of the anxiety, can help break the panic disorder cycle. And that is an example of how the cognitive behavioral model treats an anxiety disorder. We'll talk more about that in a couple lectures from now when we talk about panic disorder. Whew, I'm out of breath from talking too much. So I'm going to linger on this slide for a while. These are just some takeaways that I hope you got from this lecture. And again, if anything about this lecture was confusing, or if you just have sort of further clarifying questions, please bring them up in Thursday's class this week on the discussion board or in office hours. And then here's your further reading. You can also read any of these articles, except for maybe the neuroanatomy pictures and text. That's just more of a study guide. But read any of these other articles and react to them on the discussion board. All right. Thanks, guys. I will see you tomorrow.